Gatherings like this matter. They bring us together around ideas that have the potential to shape lives and strengthen communities. Tonight's conversation centers on one of the most compelling questions in medicine and public health: how we help people live not just longer, but healthier, stronger, and more independent lives. For generations, aging was viewed as fixed, something inevitable. Today, that thinking is evolving in remarkable ways. Researchers are asking new questions about whether the processes that drive age-related decline can be better understood, slowed, and even meaningfully altered. That shift is opening the door to discoveries that could transform health and quality of life for millions. At the Barshop Institute at UT Health San Antonio, we are proud to be at the center of that work. Our researchers and clinicians are leading nationally significant efforts in aging and longevity science, translating bold ideas into rigorous research and ultimately into real possibilities for human impact. This evening, you'll hear from two exceptional scientific leaders whose work is helping define what the future of healthy aging can look like. You'll also hear how San Antonio is contributing to research of national significance supported by major federal investment and growing momentum in translational science. Before we begin, a brief note. Following our fireside conversation, we'll open the floor for audience questions, so please be getting those ready. When that time comes, we just ask that you please keep your questions concise so we can hear from as many voices as possible. So again, thank you for being here, for your engagement, your partnership, and your belief in the power of discovery and collaboration. We're grateful to share this evening with you. And so now, it is my pleasure to welcome your moderator, Dr. Shreya Rao, cardiologist and assistant professor in the Department of Internal Medicine at UT Health San Antonio, to the stage. Please welcome Dr. Rao.
Okay, hello. Good evening to everybody. Thank you so much for coming. As he mentioned, my name is Shreya Rao. I'm a physician scientist, a cardiologist at UT Health San Antonio, and I'm fortunate enough in my role to work alongside some incredible investigators and scientists who are working to advance research that improves lives and strengthens the bridge between scientific discovery and community impact when it comes to healthy aging. It's a privilege to be a part of today's conversation and to get to interview two people that I very much look up to. So before I begin our conversation, I'd like to briefly introduce our two guests. First, I'm going to introduce Dr. Elena Volpe. Dr. Volpe is the director of the Sam and Ann Barshop Institute for Longevity and Aging Studies. She's a professor in the Department of Medicine, Division of Geriatrics, Gerontology, and Palliative Medicine at UT Health. And she's a physician scientist whose work focuses on the biology of aging, muscle health, metabolism, and strategies to improve function and recovery in older adults. Her research has helped advance the understanding of sarcopenia and practical interventions that can improve health as well as independence as people age. Dr. Volpe is also helping lead one of the nation's most ambitious new efforts in healthy aging research through the ARPA-H-funded Vital Age trial. She's also my mentor, and I've been very fortunate to spend a lot of time with her the last couple of years. Joining her this evening is Dr. Adam Salmon. Dr. Salmon is the Associate Director of the Sam and Ann Barshop Institute for Longevity and Aging Studies. He is Deputy Director for Basic Sciences, Professor of Molecular Medicine at UT Health San Antonio. His research examines the fundamental biology of aging with a particular focus on the mechanisms such as mTOR signaling, metabolic health, and interventions that can delay age-related decline. His work helps connect the foundational discoveries in the lab at the Barshop Institute to the future clinical applications that can start to improve health span and resilience across the lifespan. So together, Dr. Volpe and Dr. Salmon really represent the depth as well as the promise of the work happening here in San Antonio, specifically at UT Health. They bring very complementary perspectives to our discussion today, both encompassing the clinical and translational research space to basic biological discovery. And they together are really helping to shape a future in which healthy aging is not simply an aspiration, but an area of real scientific possibility. Please join me in welcoming Dr. Elena Volpe and Dr. Adam Salmon. Okay, so thank you both for being here, and thank you for the work you're doing to move this important field forward. To get the conversation started, I'd like to start by talking to you a little bit about your personal motivations. Dr. Volpe, what inspired you to choose the Barshop Institute at UT San Antonio and your role here in aging research in general?
So thank you very much, Shreya. This is, and thank you all for being here. It's a really nice venue and with a lot of really good people around, and I know many of you, so I'm happy to be here. So the Barshop Institute, that was easy to choose. When the position came open like 3.5 years ago for director, to replace the director of Barshop Institute that had left the state of Texas, I was at UTMB as director of City Center on Aging. I've been there for like 24 years. And I had always looked up at the Barshop Institute as one of the, beacons of the science of aging, because I thought that the advancements that the team of researchers that made up the Barshop Institute were amazing, and I was looking forward to really focus my research, which is more clinical, into translating the basic understanding of what we know about aging, what works in aging models for extending longevity and extending health span in these models. I wanted to be out there and try to move the needle and move those findings into humans and eventually population level impacts. So
that's wonderful. That's perfect. And Dr. Salmon, maybe you could speak to your story a little bit.
So actually, I came to the Barshop Institute as a postdoctoral fellow after I received my PhD training. At the time, I was very interested in aging research. It had come very recently that single gene mutations could extend lifespan in animal organisms. And I think at that time, this was just a wow moment to me, right? That you could fundamentally change the aging process by simply modifying a gene. I think there were some questions there, why would you have that gene if now knocking it out makes you live longer? And I think it just spurred lots and lots of questions about how and why we should study aging. So at the time when I was looking for a postdoctoral training, I think aging research was still sort of in its infancy, and you could find a lab at this institution or that institution that was studying aging. But really, the Barshop Institute at the time was one of the only places where there was momentum and a conglomeration of people all interested in studying aging. And what really drew me in was that I didn't have to pick a project on aging. I could work with a group of people who are all interested in sort of the same things that I was interested in. Since that time, I haven't left. I think it has been the fit for me, for the kind of research that I'm interested in, and now seeing how some of the things that we found from basic science are now translating to human application. I mean, it's an exciting time, and I'm glad it was a great fit.
You touch on such an important and kind of interesting moment we're in right now, that we've seen so much momentum grow around longevity science, around aging research. Can you maybe speak to each of you? What makes you optimistic about the field today? What maybe worries you? What are kind of some areas for concern, or where you see the most potential for new research, new investigation, and more answers?
So I think what is the most exciting thing to me is that through the work of basic scientists like Dr. Salmon and the team at Barshop Institute and also at other institutions, there's been really a flurry of discoveries of potential interventions that might, I wouldn't say delay forever, the end date, but extend longevity, and more importantly, extend healthy longevity, which is what we all aim for. So, personally, I would love to see myself, whatever is my lifespan, my total lifespan, I would like to live it to the fullest. I would like to be able to do whatever I want to do until the last day when, for some unknown reason, something happens and I'm no more. And that is what we really want to discover. So at this point, after many years, the Barshop Institute research started more than 50 years ago as research from Dr. Ed Mazur, and then it moved forward over time and became bigger and bigger, and it became an institute when Salmon and Barshop provided a large endowment to actually start it. But then at this point in time, we do have a host of potential interventions that we still need to test in humans, but we're really close to determining whether in humans they can at the very least move the needle on the health span area, because it takes longer to look at longevity, obviously, in humans. But that, and so we're going to start testing some of this health improving interventions that include not just exercise, which we always know, we all do know that exercise is good for you, is good for your brain, is good for your muscles, is good for your heart, is good for everything. So exercise, that my first thing is keep doing it. If you're not doing it, I would start a little bit. But then we have a lot of other things. For example, there's been caloric restriction, but that's tough to do because you gotta starve yourself. And then that might even, in older adults, it might actually be bad because you might actually become more frail as you starve yourself. But then there's drugs that can mimic the starvation process of a caloric restriction. And I'm talking about the drug that was first discovered at the Barshop Institute along with the University of Michigan and the Jackson Laboratories in the interventions testing program, which right now Dr. Salmon is the principal investigator on. And rapamycin was the first approved drug by the FDA that was shown in animal models to extend longevity. And then it's been followed by many other drugs. And what the thing that you asked, that the second component of your question is what worries you the most. And what worries me the most is that right now I'm seeing around in the internet, even podcasters, etc., who may tout the efficacy of these drugs in improving longevity and health span when we really don't yet have the hard evidence from human studies that those drugs are good for you and they can extend your health span and longevity. So that's why we want to do a clinical trial. So the worry is that there's some people are putting the cart in front of the horses, and so and it becomes a little bit like snake oil salesmen in some areas of this longevity frenzy. And so I would warn people to not get frantic, because we're going to have an answer within a few years, at least an initial answer in humans. We're doing at the Barshop Institute right now, there's NIH-funded program grant, cooperation grant, is a UO1 so-called, on determining what's the pharmacokinetics and pharmacodynamics of rapamycin and other, and everolimus, which is another rapalog. So that because there's no data in healthy adults. There's only, all the data on rapamycin, the clinical data comes from transplant patients who take rapamycin to prevent rejection of a kidney transplant. So, we need to know what happens to a normal person who doesn't have a kidney transplant and how safe it is, what's the minimum dose, maximum dose, all this kind of things. So we're working on that. And now with the ARPA-H, we're working where we'll have rapamycin as one of the drugs that we will test.
There were so many good pieces to that answer I want to come back to, but I want to let Dr. Salmon maybe speak to what makes you optimistic, what worries you in the field.
So I would say I am a pessimistic optimist. I think that I echo the statement Ellen made that the most exciting thing to me in aging research, I think in the last 20 years, is that we've found drugs that can extend animal lifespan, right? Simply by taking this drug, we can make them live 10, 20% longer, right? And I echo also with that. I think that gets extrapolated out into the public is this is something that people should be take. Look at these amazing effects, right? And a 10% lifespan extension would be amazing in people, but if it doesn't improve areas of health, right? I mean, that's not a great thing either. The thing about, I think, these kinds of compounds we found, it's not just rapamycin that works. We have a whole slew now, more than a dozen compounds the ITP has found that can extend mouse lifespan, right? They target multiple different pathways. Things like mTOR, things like glucose metabolism, things like inflammation, right? So aging's complex. And again, 30 years ago, I think it was science fiction that we could even target the biology of aging. But it's science fact right now. We can do this, right? And I think that there's still a long way to go, as Ellen has said. But I think the most optimism I have on that is that we could actually go in and repurpose these drugs, not just to extend lifespan, to improve health, right? Potentially even overall health, not just targeted disease. Many of these drugs are already used for disease. But if you could take this drug now for off-purpose target for a disease or a condition or a pathology that there's nothing we can do now, that's an amazing thing, right? And so I think the cautious interpretation is that there's the potential. I think also too, many of these drugs that we found, actually none of these drugs that we found can rejuvenate, right? Except maybe in certain instances. So if you've come into the clinic, I imagine, I'm not a clinician, you come into the clinic and you're already having these conditions, it's very, very, very hard to reverse them. We could potentially delay them, and I think that's also important. But the concern of thinking that I'm becoming old, I'm becoming sick, I'm becoming frail in different ways that we can change that immediately, that's not there yet. It may be in the future, but I think we're at those steps where we can now start to test those ideas. And so I think that's where my caution is over-interpretation, but it's a very optimistic time too.
Yeah. You both touch on this idea of a paradigm shift in medicine broadly, and definitely in the space of aging and the care of older adults, where we go from thinking about how do we treat a disease and be very reactive to how do we be more proactive? How do we think about bending trajectories of aging and aging-related decline before they start? Can you both touch, and maybe Dr. Volpe, you could get into more detail. You mentioned what goes into healthy aging and health span, and you made that distinction of health span versus lifespan. Can you talk more about what that means to you as a clinician, to you as a physician scientist, when we're measuring this in humans? What are some indicators of health span, and how do we conceptualize that?
Yes, a good question. So the health span is essentially the years lived in a disease-free and in full independence. So if we can maintain our independence and our health as much as possible, that's what health span is. How do we actually operationalize the measurement of health span? That's a big question, and we're still struggling with in the ARPA-H-led PROSPER consortium, of which we're the biggest part because we have the largest contract with ARPA-H, but there's several other places that have contracts with them to actually better define what is this health span. So one measure that has been brought forward recently by the WHO is the concept of intrinsic capacity. Intrinsic capacity is this concept by which each individual has the capacity to live, work, function well, and they conceptualize it like this general idea of having 5 major realms of function that we can look at, and those are locomotion, which is moving around. There is obviously cognition, so being mentally sharp. There's psychological health. There's another area that's very important. There's sensory, so vision, hearing. And there is what they call vitality, the WHO calls it vitality, which is the mixed pot of everything else. So it's cardiometabolic health, so cardiovascular health, diabetes, metabolism, thyroid, whatever you want to toss in there is all in there. And so to measure that, we are trying right now as part of this ARPA-H effort to have to come up with a scoring system, a measuring and scoring system for each of those 5 domains of intrinsic capacity, so that then we can measure it over time and we can look at those at this intrinsic capacity, how it changes over time, and how the interventions, in our case, the drugs that we're going to use over a significant long stretch. Our study is going to be 3.5 years long with drugs taken for 3 years. So it's a long clinical trial. And so we want to see how these drugs, if and how the drugs can slow down the natural decline of this intrinsic capacity that happens over the years. And so we're going to take people in their 60 to 65, 60 to 70. We're still negotiating with ARPA-H about that. We're trying to expand it to 60 to 70 years old at the very least, and then start the intervention and then monitor this intrinsic capacity over time to see whether any other drugs can really impact it significantly. And so that is otherwise my personal way of looking at health span is doing what I want to do for as long as I can without having the impediment of having problems with my mind, with my body, with my muscles, with my bones, my joints, and all that kind of things.
Exactly. We use the terms disability, frailty. We use a lot of these terms, and we don't just talk about preventing disease outcomes. We talk about really trying to prevent what we think of as the natural process of aging, right?
Yes. And the critical thing is that, we think about aging, the scariest thing that a lot of people are scared about, and reasonably so, and rightly so, is Alzheimer's disease and cognitive decline. But there's just one portion of aging. It will affect, 15-20% of the older adults. They say that it's going to increase the percentage of people who will be affected, but the reason is that we're getting older and older, and so as we get older, obviously age is going to increase the risk of developing it. But that's only one part. 80% of older adults above the age of, say, 85 are functionally, they have some functional limitations, and that's a lot of people. Much more so than, say, cognition. And that means that those people are essentially relegated to their home or their assisted living facility, nursing home, or whatever. And so those are those things that, that honestly, personally, I'm more concerned about developing a physical disability that will not permit me to go around about and about than a cognitive disability, because that, to me, after the initial problems, I will be well gone mentally, and it's going to be my kid's problem, not my problem. Whereas you know, if I am cognitively intact, but I can't go around, that's a big, big issue. Maybe my husband's problem too. Yeah. 'Cause, yeah. So that's, you know, that is what so that's why I focused a lot more on sarcopenia, muscle loss, functional loss, physical function, recovery from, say, hip fracture, those kind of things that are really, that can really cripple someone.
I'm glad you brought up that last piece because that's been a huge component of your work, right? Recovery from a major life event, which the frequency of these increases as we get older, and that recovery time really varies from individual to individual, and that seems to be something, a potential lever that we can intervene on.
Yeah.
Dr. Salmon, can you speak to, you mentioned the complexity of aging and of aging biology. What pathways, you know, you mentioned inflammation, you mentioned metabolic health, and we've talked a little bit about the calorie restriction pathway. Can you talk about what are the pathways that you think are most promising? And are these pathways distinct? Do they share some common, you know, shared mechanisms? Can we learn something from all of these different candidate drugs that you've learned about in the ITP?
Yeah, I mean, so about 10 years ago now, a consortium of people came together with this idea that there are these hallmarks of aging, right? And so they've published now a few different studies, and they kind of fall in things that we know change with age. Biologically, we get increased inflammation, we get decreased DNA repair, we get mitochondrial dysfunction. I mean, I think the interesting thing is that all of these really interact. So I would argue that what we are seeing is that there's no real central player that we can pick out from these. I mean, I have my own pet interests on things that I think really drive it, but I think it doesn't invalidate the other areas, right? And for example, something like mitochondrial function. The mitochondria, anybody remember from your basic biochemistry? It's the powerhouse of the cell, right? It makes the ATP, it keeps the cell in homeostasis, keeps it functioning normally. In my mind, if that goes away, everything falls apart, right? And so the structure to maintain those types of things, cellular organ homeostasis, is critical to maintaining, you know, health overall, but health with age too. And I think what knowing, you know, I think these hallmarks of aging have given us sort of targets to actually address some of these questions about complexity, right? It may be that frailty is really driven by one of these pathways, or cardiovascular disease is driven by another set of pathways, but not so much these ones, right? And so now we could dig into sort of personalized approaches. If you're at risk or you currently have any of these diseases, maybe we could target that rather than here's this overall, everybody should always take rapamycin or everybody should undergo calorie restriction. There may be ways to actually target what you're at risk of or what you're suffering from, you know, and actually improve that, right? And this is really, I think, the idea behind that concept of geroscience, which again, about 10 years ago, this was conceptualized. It's really the blending of biology of aging with clinical application, right? Understanding what are the targets of aging or the mechanisms of aging to develop targets and actually improve health, right? And so, you know, in theory, what that means is that we could then go in, develop new opportunities based on those. And so I think that's where, rather than for me picking a specific mechanism, I think is most important. I think figuring out how those mechanisms drive the various aspects of aging, the sequelae that you might see towards disease, and using that as a better interpretation for informing for clinical practice or how you might approach improving health overall. Again, it comes back to can we, are we delaying aging? I think very, very important. Or are we treating disease? And it might be two different pathways that we have to do this.
That's a really helpful, I think, moment to segue into the ARPA-H trial, which you've talked a little bit about, Dr. Volpe. Maybe you can introduce the trial structure, what it is, what we're trying to achieve, as well as some of the candidate therapies that we've entertained and that are really informed by what Dr. Salmon just discussed.
Yes, so this ARPA-H opportunity came about last year, and we thought we would be the right place to actually do the clinical trial for repurposing drugs to try and improve health span in relatively healthy people. So, in other words, what ARPA-H, which is, by the way, a brand new federal agency that was created by President Biden a few years ago, and then it's still ongoing and still has quite a bit of money. It's from the Department of Health and Human Services, so it's not NIH, it's a different branch. And they want to really accelerate clinical research as fast as possible to bring interventions to fruition for the general population. And so the way we approached this ARPA-H trial was to say, okay, we're going to do a large clinical trial. They dictated, ARPA-H dictated the general population. We had a age range and more or less health status. And so, and we decided to which drugs we wanted to test based on the information that we got from the interventions testing program and from post-market information for some of the drugs that we selected. And so the drugs we selected are rapamycin, obviously. We selected semaglutide, which is one of the GLP-1 receptor agonists. It's Ozempic when injected, or Rybelsus when taken orally. Now they have oral Ozempic too. And then dapagliflozin, which is Farxiga, which is a drug. So 2 diabetes drugs and 1 rapamycin, which is an immune suppressant. And the 2 diabetes drugs were selected because the class of drugs of Farxiga, dapagliflozin, actually has been shown by the interventions testing program to extend longevity in mice. Farxiga per se, not yet, but it was, I think it was Jardiance and dapagliflozin. No, canagliflozin. Anyway, so we're saying, okay, let's try with those, because we do know from post-market data from Farxiga, from the SGLT2 inhibitors, which is the class of Farxiga, canagliflozin, and the others, we do know that they reduce cardiovascular risk. Farxiga also reduces renal risk. Ozempic or semaglutide also reduces cardiovascular risk in diabetes, and now is obviously used also for obesity treatment. So, and then rapamycin, as I said, is the first and the most famous, and some people are taking it off label because their doctor prescribes it to them. And has this immunosuppressant effect, but what it does also reduces mTOR signaling. So usually as we get older, mTOR, which is this important fuel switch in the cell that is critical, its activation is critical to, say, grow muscle acutely after a meal or after exercise, but if it stays upregulated for a long time, then it can create a lot of problems, increases insulin resistance, so it tends to induce type 2 diabetes. And then it can increase the risk of cancer. It can increase, so there's a host of diseases that can come from hyperactivation of mTOR. So we're looking for a dose that doesn't completely suppress immunity, because we don't want to do that. What we want to do is just get enough to just lower a little bit mTOR and then improve the outcomes for the people who are taking it. And so those are the drugs that we're taking. The other thing we're going to be doing, we're going to study a lot of people. The target is between 700 and 1,000 people. We're still negotiating to see if we can find additional funding from the drug companies that make the drugs to expand the cohort. And then the other thing we're going to do, we're going to try to go in our community. We will do the entire trial here in San Antonio and South Texas. And we want to go out there and have sites in the community. And so while Barshop has its own clinic, and so we have our site in the Barshop Institute, We also are buying right now a mobile clinic to go around in a radius of about 80 miles, because we want to be able to come back home every day, to go and reach out to those people who are more rural, who normally don't participate in clinical trials, because those are the people we're trying to target in general, along with the people who go see their doctor at UT Health. But we want to see the people who don't see the doctors at UT Health and then go to their own little clinics outside in the community. We're also going to have a clinical site at the food bank in West San Antonio, because again, we want to reach out to people who normally don't participate in trials. We will utilize the University Hospital clinic, the Texas Diabetes Institute, which is downtown. And so trying to reach out as much as possible in the community, in San Antonio and surrounding areas, because we want to be the results of this study to be as impactful for the general population that we have in San Antonio, including those who are not normally participating in trials. And that's bad, because we need everyone to participate in trials if we want to generalize the results that we get.
And I'm going to toot your horn a little bit here, but as far as I know, this is the largest human study clinical trial for prolonging lifespan and health span to date in the world.
Yes.
Yes.
So far, we're going to be, unless someone else is coming around.
And this will be
Next couple of
led here.
days.
This will be led here by our investigators at BarShop in San Antonio.
Yes.
So I just want to highlight what a big achievement that is, and
Yeah.
Dr. Volpe led
Yeah.
the team on all of that. But it's going to be an incredible couple of years. So I think that kind of leads to the next and our last question before we break for audience questions. Dr. Salmon, what makes you what impacts are you most interested in seeing in your career over the next 5 to 10 years? What do you think this next decade looks like?
I think this is a big step, right? My career as a basic biologist has all been animal models. Animal models are great for studying basic mechanisms. For aging studies, I think researchers fall into two sort of camps. One of, if I do something to this animal, does it live longer, right? End of life is a pretty easy thing to measure in theory. Or if I do something to this mouse, does it make them healthier in some way? We can do all these things. I think the real challenge is trying to bring that to an understanding of how we can help people, right? I mean, if you're looking at a mouse and we do some assay on it and it looks like muscle function has declined, does that mean anything about human frailty? That's a real challenge, and I think we overlook it a lot. The other thing is that when we're looking at an aging animal model, they are not dying of the same diseases we're dying of. A mouse is dying of cancer, almost exclusively cancer, which, this room knows we die of lots of different things as people. But I think more importantly, in animal models, we really can't get at quality of life. Right? I think this was a point you raised at the start, thinking about how we improve quality of life. We can't ask an animal how it's feeling today. We can't look at pain very easily. We can't do aches and pains and everyday things, even things like back pain, right? How many people have back pain as they age? Really, it's a function of a standing bipedal, standing on 2 legs. A mouse doesn't get back pain. It's on 4 legs. And I think as a basic biologist, we don't often think about that, those quality of life things. We are using the wrong tools to understand how to help people. I think we are getting better at that, but we still have that problem. So to me, the most exciting thing in the next 5 to 10 years are actually testing the things that we've been looking at for now 20 years in animals. How do they help people? And again, I don't think that this is pie in the sky kind of thinking that we can help people. We can, and we know we can with things like exercise and diet. We think we probably can with drugs. Implementation, the next 10 years, that's going to be really tough. How do we get people to really do these things and improve their lifespan? But I'm excited about it, and I think that you know, the fundamental question, can we make people live longer, really should just kind of go away. Can we make people healthier for longer should really be what we're asking. And I think clinicians think that all the time. I think basic scientists do not think that. And what I've really enjoyed about how BarShop has evolved over the years is that we've had those connections build, that we're able to start thinking about that I don't just sit in the lab and go, yes, we can do these things. We can actually think about how we can improve people's health. So that's my excitement in the next few years.
I love that. Dr. Volpe, what are you looking for in the next 5 to 10 years of your career, and what do you hope to see the field evolve as in that time?
Well, I hope we can bring this trial to fruition to begin with. That's at the top of my mind right now. The trial will launch probably early next year. We're in the working phases right now. We're setting up the sites, setting up all the background things that need to happen. You know, get the staffing, get all the important things, and train the staff, and get, you know, approvals, regulatory approvals from our institution. And you know, the institutional review board, and then from the FDA if necessary, and from ARPA-H and all this kind of things. There's a lot of barriers and hiccups that need to be addressed before we start recruiting. But when we start recruiting, we'll let you all know, because when we finalize finally the inclusion-exclusion criteria, I will try to keep the exclusion criteria to a minimum, because I think it's important to get people involved that are the real potential users of the drugs, and not just some super healthy group of people that normally gets assigned to clinical trials. So within my capacity and the team's capacity, we're going to try to keep it as open as possible. And then, you know, we're also starting discussions with our concierge at UT Health and the executive health. We're trying to also open a healthy longevity clinic in the BarShop Institute, or or have some of our clinicians. You're one of the clinicians. Dr. Tiffany Cortez is down there in the audience, and she's actually the medical director of the BarShop Clinical Research Unit, and we're working together to set up a system where we can provide evidence-based interventions for people. Right now, in the absence of hard evidence like clinical trials, but based on what we know from preclinical studies, what we know of the patient risk factors and current diseases, try to perhaps be more mindful in terms of, utilizing the drugs that those patients need for their diseases, but utilizing the drugs that we know may improve also longevity and health span in animal models. For example, if you have, people with diabetes, we do know that SGLT2 receptor inhibitors are potentially good for longevity, so why not trying to use those? GLP-1 receptor agonists, if tolerated, are great and make people lose weight. They give you that little bit of caloric restriction type of thing, like naturally, because so why not utilizing those and so forth. So even for those who have cardiac problems, SGLT2 inhibitors are good for, in the congestive heart failure. So why not using those instead of another diuretic, and so forth. So having a clinic that is not a snake hole clinic, but is a clinic in which we want to provide the care based on the evidence of what we know preclinically and what we know of the patient specifically, so very much personalized. I believe in personalized care. Pretty much all geriatricians believe in personalized care, I think, because each patient is different. Each patient needs different things. And while I wasn't born a geriatrician, I became one by trade. And so I was an endocrinologist to begin with. So I was treating diabetes, pretty much cookie cutter type of stuff. And over time, I've learned that really you need to adapt. And each patient is their own world. And so you gotta see the patient as one and not as, like an algorithm. That, algorithms are good, but not always. And so that's where we want to go.
Thank you, Dr. Volpe. So I want to open the floor for questions. We want to fit in as many of your questions as we can. Just a reminder, please try to keep your questions brief. We're going to be coming around with the microphone. Please wait for the microphone so that we can make sure all of our speakers hear you. I think we have one in the back.
Thank you. About the clinical trial design, have you decided yet whether all of the study participants will take all 3 drugs, or will they only take certain combinations, or perhaps it's not decided yet?
No, that's decided. It's going to be single arms. So there's going to be 4 arms. We're going to have 3 drug arms and 1 placebo arm. So the placebo is important because we need to see the decline in a group of people if we want to compare it to what the drug does. And so we want to have that. And it's mandatory, really, if you want to eventually get the drugs repurposed officially by the FDA for different indications. So it's going to be separate arms, as far as I know. And so far, that's the plan, and we're powering it for that.
It seems to me that we have a unique advantage in San Antonio. Where do you think that we fit in the spectrum of science and understanding of aging at the Barshop and the Brain Health Institute? What is our unique advantage and where do we fit?
I would like to ask Dr. Salmon, because he's the incoming president of AGE, the American Aging Association. So he just got he's just starting his term next week. so he's the national leader.
I don't know about that, but
Yes.
I think that San Antonio has been building momentum for now more than 20 years as the Barshop Institute officially, but 50 years with aging research as really the focus. I would say I think San Antonio doesn't get the recognition, and if that's the right term. I think that, oftentimes when you read a story in the lay news, it'll be from somebody else that's put together this press announcement of they've fixed aging or we're going to fix it in 5 years. I think where our solidness has been is that we built that foundation, and I don't think any of our researchers at UT Health would let these things go out, right? Unless we're very, very sure of them. And I think that's where I come back to my pessimistic optimism. If we find something extends lifespan in an animal model, improves health, we want to be really, really sure about that, right? I think that's that next application. So to me, it's been just sort of this critical mass over the years. We've had a lot of researchers here. I think we've worked really well together, and that may be part of the culture of San Antonio too, is that we have no problem collaborating. I might work with Shreya on building something that she's coming up with the basic science side, or vice versa. So I think that's really where our added advantage has been. And we've been able to resource the institutional support to get these types of studies going, like ARP-H, like animal studies, looking at novel
When can we shout, we're number one?
We
can shout it now.
We are. We can shout it now because we are the only institution that has 5 of the 7 program grants that are competitively given away by the National Institute on Aging. And they are the Nathan Schock Center for Excellence in the Biology of Aging, he's the director of it. There's the Claude Pepper Older Americans Independence Center, which is the translational center, which I'm the director of. There is the center, there's the Alzheimer's Disease Research Center, which Dr. Seshadri has. There is the Center for the Demography and Economics of Aging, who Dr. Rebecca Wong has. She came with me from Galveston. And then there is also the minority aging research. It's the Resource Center for Minority Aging Research, which is another large P30 grant from the NIA, which Dr. Fernando Rios Mena at UT San Antonio is the PI of, and he's an adjunct faculty in the Barshop Institute. So now that we're together is even better because we have all this 5 things. Nobody else, not even Harvard, not Yale, not Princeton, not Buck Institute, none. That we're the only one who have everything. So I would say we are the top Ivy Leaguer here in aging research. We have it here. Maybe we want to stretch it a little bit, the Ivy stuff. Yes, so, and last year when I pulled numbers from the NIH for what just the health science campus, I didn't even include a UTSA at that time, for what we bring in terms of dollars from the federal government, from the NIH. Last year it was $53 million a year of grant dollars that we were bringing in. This year, I don't know. But at that time, I also pulled numbers from UT Southwestern, UTMB, UT Houston, because we were sending a grant together, which then we got. And we were the top of all 3 of them, including Southwestern, which has got a big research enterprise. And we're better than them in aging.
I want to add on.
So,
I want to add on, and that's such a positive way to end that, but I wanted to add on one comment about San Antonio specifically, because I think separate from the academic infrastructure and everything that Dr. Salmon and Dr. Volpe mentioned, I think it's worth mentioning what's unique about San Antonio as a community. This community, and this has been a challenge, obviously, health-wise in this community, has one of the highest burdens of chronic diseases in not just the state of Texas, but nationally. And what we're seeing in terms of the demographics of this community, in terms of diabetes rates, heart disease rates, high blood pressure, high cholesterol, these are demographics that we expect the rest of the country to hit in the next 30 years. And so studying what works in our population and understanding the science of that here actually helps not just inform local solutions, but really prepare us for what's coming nationally to the rest of the country, and then really ultimately globally, what we're going to see all across the world. So it's not just a valuable opportunity, it's an imperative to be the leaders in this space here. We have time for one more question.
Well, this is wonderful, and thank you. I think all of us in this room are looking forward to a longer health span from your research. But obviously it's very complex, and I was interested in things like you're looking at 3 different drugs in this trial, and obviously each one has different possibilities. And you already pointed out that there are so many different systems that can be affected. So I'll just ask about rapamycin and say, what do you think the most effective, what system do you think rapamycin will be most effective on? I mean, it's a molecular mTOR regulator, so is it going to be cardiovascular or cancer or brain, or what do you think is the area that rapamycin will be the most effective in?
Well, I could say from the animal models, rapamycin, I think we at the ITP kind of stumbled on a gold mine. It does seem to target many different areas. It seems to, in some ways, rejuvenate cardiovascular function with age. In mice, this is all animal models. It seems to prevent some types of cancers. It seems to improve brain health. But there are some things that be very, very bad at, right? Because it is as Elena mentioned, it's the regulator of growth. If you were a pregnant woman and you wanted to have a child, it would be a very bad choice, right? Other things like wound healing can be effective. But what we do know too is a drug like rapamycin, most of these effects go away when you stop taking it, these negative side effects. So wound healing improves immediately after stopping rapamycin treatment. So I think we're at the stage in animal models where what we've done is essentially given a drug for the rest of that animal's life and looked at its effects on life and health. But I think in people, there may be a whole different solution there, right? Where it might be something where you take it for a short period of time to improve some aspect, and then it's sort of a resetting period. We don't know that yet. I think it would be exciting that we could do something like that. I mean, ultimately, again, I'm the non-clinician on the panel here, but I think it's something to discuss, with your provider at some point. You might be a better candidate for one of these other drugs based on your past history, based on your genetics, based on the fact that you're a woman versus a man. And this is what I think what's exciting about this stage is we'll figure some of these things out with this ARPA study.
Yeah, so I think to add to what Adam just said, I think that the critical piece, right now we'll have to do a trial that is the classic, you take a group of people, randomize them by the flip of a coin, and they end up getting whatever. But the future, in my view, will be to personalize care as much as possible. And that's, precision medicine, there's all these new words that, but in reality, a lot of doctors do already precision medicine based on what they see in their patient, how they respond to drugs, or how bad some drugs can be for them compared to others. So we want to do that in a more knowledgeable way, so that if there's a way to determine how someone responds to one drug versus the other, we want to know that. There's now a few drugs for which we know if we need to change the drug or increase the dose or decrease the dose depending on what your genes are or genetic makeup is and so forth. But that needs to go much further, I think. And eventually we'll have to, and hopefully for the future, maybe with some help also from AI, not using it, just using it, not being slaves of it, but using it the right way, possibly we may be able to end up figuring out more rapidly which drugs are most effective for who.
So with that, we're going to close the question session. I know we have a few more questions. Please hold on to them and approach Dr. Volpe, approach Dr. Sandman, or any of us afterward, or if you send them to us, we'll be sure to get in touch with you and provide more answers in more detail. We just want to make sure we keep on time today. I do want to thank Dr. Sandman and Dr. Volpe for their time and all of their expertise in sharing so much of their experience with us today. I can say from my personal experience, I am early in my career, and it's been a very happy accident to get to walk into a situation where I get to work for someone like Dr. Volpe and learn from Dr. Salmon and really get this opportunity to be part of this extremely exciting moment. As you've seen today, hopefully we've given you a sense of, we really feel like the future of healthy aging of this field is being written here in San Antonio. And we think this is a really exciting moment for all of us and for the future of this research. So we thank you for joining us. And like I said, please come to us with more questions if you have them.
Thank you, Shreya.